Hello, my name is Chelsey Dunham, and I'm a postdoctoral research fellow at the NIH, and I'll be presenting my work titled Limited Gender Differences in Gastrocnemius After Single LIPUS or pFUS Treatment. Therapeutic ultrasound, like low-intensity pulsed ultrasound and pulse-focused ultrasound, are used to enhance healing of musculoskeletal injuries. Acoustic radiation force or pressures from these sonication treatments drive cellular changes, including the activation of sodium-calcium channels, hyperproliferation, and increased cytokine signaling. Previously, pFUS was shown to upregulate cytokine expression in the hamstrings of control and dystrophic mice. However, this work only evaluated changes in female mice. Therefore, the objective of this study is to evaluate gender-based differences following single LIPUS or pFUS treatment to healthy gastrocnemius. Male and female C57 black 6 mice that were 6 weeks old were used in this study. The right gastrocnemius of these mice were treated with single bouts of LIPUS or pFUS. Control mice were not treated. At 1, 8, and 24 hours after treatment, muscle was harvested and evaluated for cytokine expression. At 24 hours after treatment, muscle was also harvested for histological and immunofluorescent staining. For histological and immunofluorescent staining, 24 hours after treatment, muscle was harvested and mounted in tragacanth gum that was flash frozen in liquid nitrogen-cooled isopentane and cut as 10 micrometer transverse sections on a cryostat. Female muscle will be displayed in the top row. Male muscle will be in the bottom row. The first column is the control group, which received no treatment. The second column is 24 hours after LIPUS treatment, and the third column is 24 hours after pFUS treatment. These H&E stain sections are used to demonstrate that there's no change to the macro level muscle structure as a result of gender or 24 hours after single ultrasound treatment. I then stain for laminin, which is the protein part of the basal lamina of the myofiber. It's typically used to measure myofiber cross-sectional area. Female groups are shown in orange, male groups are shown in blue. A 2A ANOVA demonstrated that there was a significant effect due to gender. However, there are no significant post-hoc Bonferroni comparisons demonstrating that there's no change to myofiber area as a result of gender or single ultrasound treatment. I also stained for collagen, which is the protein part of the reticular lamina of the myofiber and is typically used as a measurement of extracellular matrix deposition or muscle fibrosis. You can see that there is no change in ECM deposition as a result of gender or 24 hours after single ultrasound treatment. For cytokine expression, muscle was harvested at 1, 8, and 24 hours after treatment. It was immediately flash frozen and homogenized with protease inhibitor cocktail. The total protein was quantified using a BCA assay, and the cytokine panel was loaded at 2 mg per ml for each sample. The data will be displayed as a heat map where expression is normalized to the average control expression. If you see an asterisk, that means that there is a significant difference due to the gender matched control. A G means that you have a significant gender difference at that time point, and a number means that it's significantly decreased due to the gender matched number that's listed. An X through a box demonstrates that that cytokine was not detected. The data is displayed in columns where you have female on the left and male on the right. You have your 1 hour time point, 8 hour time point, and 24 hour time point after treatment. This first group of data is for LIPUS treatment, and the second group is for pFUS treatment. If we look at the general macro level statistics, specifically looking at differences that occur solely to time and gender, you can see that the same number of differences occur for both for LIPUS. However, for pFUS, we actually have more differences due solely to time than gender. More importantly for this data set, when we look at the post-hoc gender differences, you can see that we have a total of 60 potential gender differences for LIPUS and 48 potential differences for pFUS. The differences in numbers here are a result of the number of cytokines that were detected in each study. However, looking at the results, you can see that only 1 out of the 60 potential gender differences for LIPUS and 2 out of the 48 for pFUS were actually significantly different, demonstrating that gender is not significantly directing or impacting cytokine expression in muscle. Looking more closely at the cytokines themselves, you can see that LIPUS and pFUS are both regulating VEGF, where you have an upregulation at 1 hour and downregulation at 24 hours. Looking specifically at lipus, you can see that IL-15 expression is increased at 1 hour. Looking specifically at pFUS, we actually have decreased expression of IL-2, IL-9, MIP-1 alpha, and IP-10 at 1 hour. However, at 8 hours, we have increased expression of IP-10 and MIG, and at 24 hours, we have increased expression of MCP-1. So overall, what this is showing us is that we have an early proangiogenic response in both treatment types. However, with lipus, we also have an inflammatory cell recruitment signaling or pro-inflammatory response signaling at 1 hour. But this same signaling response is delayed with pFUS as it's upregulated at 8 and 24 hours. Looking at cellularity, specifically macrophage M2 population, you can see that I have stained for laminin, which is that basal lamina protein surrounding the myofiber, to help identify these cells as they should be located within the interstitial space between myofibers. I also stained for F480 as a general macrophage marker and CD206 to identify the M2 macrophage population. The M2 macrophage population is double positive for F480 and CD206 and is denoted with the white triangle, whereas only F480 positive cells are denoted with the white asterisk. Looking at total cellularity, you can see that there is an effect due to treatment and gender. However, we have no significant post-hoc Bonferroni comparisons due to gender. We do have a significant increase in both male and female 24 hours after lipus treatment compared to control. Looking at the M0 macrophage population, so this is F480 positive, CD206 negative, you can see that there is a significant effect due to treatment and a significant interaction of these two factors. For the first time, we actually do have a significant gender difference, and that's 24 hours after lipus treatment, where the female are significantly increased compared to the male. Looking at the M2 macrophage population, so this is our double positive F480 CD206, you can see that there are no significant differences. So overall, this data is telling us that 24 hours after lipus treatment, we have hypercellularity, but it is not due to an M2 macrophage infiltration. So overall, this study has shown that there are limited to no gender differences in muscle macrostructure, cytokine expression, and cellularity. Looking more detail at macrostructure, including myofiber area and ECM deposition, there are no differences 24 hours after single ultrasound treatment. Looking at cytokine expression, we had an early proangiogenic response with VEGF upregulated at 1 hour for both lipus and pFUS. We had inflammatory cell recruitment signaling that was upregulated at 1 hour for lipus. However, this was delayed for pFUS and upregulated at 8 and 24 hours. For cellularity, we had hypercellularity 24 hours after lipus treatment, not as a result of the M2 macrophages. Therefore, this means that other cell types likely contribute to the cellular influx, including M1 macrophages, neutrophils, T cells, or PEC7 positive satellite muscle stem cells. Another thing that's of interest is that pFUS likely presented no change in cellularity due to its delayed onset of its pro-inflammatory response. So this is something that we could assess by evaluating cellularity at 48 or 72 hours after treatment to just see if this delayed pro-inflammatory signaling leads to a delayed hypercellularity in pFUS-treated muscle. Limitations of this study include that only one time point evaluated muscle macrostructure and cellularity, and we only evaluated healthy tissue. Future work includes using flow cytometry to evaluate immune cell population infiltration longitudinally after treatment, as well as assessing treatment strategies following muscle injury. Thank you so much for your attention, and I'll be happy to take any questions. Please contact me via my email listed on the slide.